read this paper? Okay. Let's call ISME 1. Then, who read this paper? Then, who read this paper? Ok, 4. Cos'è? EMS? Così? Sì. Who read this paper? Okay, this is the most popular. 10. I mean, it doesn't need to be precise. And who read this paper? 2. Okay. 3. 2.5. Okay. So let's try to. How is it?
Group here divides in 2 and explains to. So the plan is that. Yes.
No, it's just because you divide into 2. You're going to be like this 2.
Yeah, no, I think we can divide in more groups.
More groups. The idea is that who read something explains to the other, so that everyone in the end knows what we are talking about.
So you have an idea. So let's do the groups such that there is someone that read a paper in each of the group. Yeah, exactly. So who are the 3 people that read this paper? 1, 2. Why you are raising the hand?
No, I raised it partially.
Okay, but okay.
But did you read something else?
Yeah.
Then,
Okay.
then you count for the other.
Okay. So let's do other.
Let's pick this one or this one.
Okay.
We have 4 groups.
Okay, this one is more important, I think. So who are the 4 people that read this paper? 1, 2. They were 4. I'm sure. I counted very carefully. 1, 2, 3, 4. 1, 2, 3. Who is the 4th? You read it? Yeah, so 1, 2, 3, 4. Okay. So then these 4 people go in different corners of the room. Okay? And then the others divide more or less randomly. You do a poll, you check if all the papers are represented, more or less. And then you start discussing and creating a common knowledge in the group of what you read.
Okay?
Is,
self-organize.
So, and we do like, I would say 20 minutes. The idea is that 25. But we do 20, Martina, we say 20 and then we do whatever we want. We do 20 minutes, 5, more or less.
No, plus.
And the goal is that each of you read at least 1 paper. So the idea is that you sort of say what is the important message of that paper. You start discussing with the idea that you find, so let's say the output that you'd like to have are questions. So not questions for us, but questions that you think emerge from these papers. So what are the important questions that after reading these papers you think are interesting to address as research topics.
As a discussion for the rest of the time.
Yes. Okay.
Self-organized.
So, yeah, self-organized. So of course, I mean, it's not like that in these 20 minutes we are gonna lay like this. So if you have, so the way to use this time and us in these 25 minutes is that if you have instead specific questions. So I didn't understand. We as a group are not understanding this particular thing. The moment to discuss these things is these 25 minutes, okay? With the goal of arriving at the end of the 25 minutes, which are gonna become 30, with a list of questions, okay? Good, so the 4 people of this paper go in the corners and the others, yeah, raise the hand. You can go to another room, yes.
Whatever you want.
Go to another room. That's a great idea. But yeah, the important point is make sure that out of these 5 papers, at least 4 are represented, right? So that you have like you have not read only one paper, all of you. So. Yeah? Huh? Let's do 7 more minutes. So at noon, 20 past noon. Try to focus on formulating the questions. Okay. Okay, is everyone back here, or there is someone? Ah, you are the ones. Okay, good. So I think we can start the discussion between groups. So as we said before, the goal is to try to identifying questions that are sort of broad and big in terms of their importance. So I don't know, perhaps each of the group can report a little. I mean, we saw that there were some discussions in the sense that there was disagreement sometimes within the groups, which is a good thing. So I think that you can sort of start to report each group individually. What are the things that you think that are critical? So the points that were sort of the most controversial or the most unclear or the most interesting, which usually these three things converge. So who wants to start?
Okay, so for us, it was very interesting that we can see very clear that the community ensembles in a genomic level. Maybe it's not that clear in big organisms, but here it's very clear that you can lose genes because some other has the gene to produce something I need. So this kind of interaction is very deep, and you get to a neutral connectivity in the whole community. So that's our first impression, let's say. We have questions, but let's continue with impressions.
But, okay.
Somebody else?
Does someone have say about this?
Yeah, me too, but I think there are two Microsoft
Here?
microphones.
We were discussing about exactly this idea of a genomic community ecology, and we were thinking like you're really thinking about this because at least for me, I've studied ecology before, but not in this sense. It was because my microbial ecology, the genomics is the main tool, and coming from that other side, when you look at it and you're making all these deductions from the genome, how good are those extrapolations? Are they fine or not? We talked about this specifically about the copiotrophic, oligotrophic article using the genomics to establish which bacteria is from which strategy, and how good is this extrapolation?
Do you want to say something, or okay? Yeah.
Again, about the same paper, we were discussing about how counterintuitive some of the genes are with the oligotrophics and the copiotrophic ones, because, for example, we had like a more robust sensing of the environment and of nutrients in the copiotrophic ones. There's the ones who are in the better condition, and we was expecting the opposite, that like the ones that are in a poor environment would sense better where to go and where are the nutrients. So we find that some genes, gene profile of this metagenomic thing was counterintuitive.
So just to be sure, so the counterintuitive aspect is that who lives in the best environment, so like copiotrophs, are better at sensing, while you would have expected that oligotrophs are better at sensing because they are in a...
Yeah, but it was an example, but we have the motility thing as well, and some of this.
Okay.
Of motility is mostly related to the fact that you find copiotrophs on particles. So the distribution, even when you are in a rich environment, nutrients sometimes are aggregated in marine snow and particles. So the idea is that if you have flagella, or you can move and you can sense the environment, you're going to reach these nutrient patches. So the advantage why copiotrophs would actually need flagella is that they are looking for these nutrient patches, while copiotrophs in these potentially very high nutrient environments, they don't thrive. This is a possible way to think about it.
But I think that, okay, yeah. I mean, but I think that the point is how you look at them. I mean, well, okay, I'll say that later.
One thing that I was thinking now is like about this thing of attaching into particles. A copiotrophic bacteria is copiotrophic because it does this, or it's the opposite? It does this, so it is a copiotrophic. Like, what's the first?
I think it's a bit the chicken egg. No, yeah.
Yes, because it's like the other could do this, could do the same, but it does not. So, okay,
Yeah.
I'm good.
But related
Because
to
I think the definition of a copiotrophic is kind of, it was a problem to us because it's because of the environment and the functional things, like physiologic things. So, like, it's a copiotrophic because it's found in an environment that has a lot of nutrients, or is a copiotrophic because it has some type of physiologic functions or whatever. It was kind of difficult to understand.
Yeah. We don't have them.
Divide something that is a spectrum or a continuum into categories. But the idea there, I think that it's both. So you find, if you look at community structures along gradients, for example, they find that you have these changes. And then you say, okay, what differentiates these bacteria that I find in this environment from these bacteria that I find in another environment? And then they look at physiology or genomic features, and they say, oh, those are different, apparently. But again, and to reconnect a bit to this broad theme, I would say that it's not always true that what you have in the genome is actually what you are doing in a community. So because the genome is telling you the potential, so you can do a range of things, but then what you are exactly doing in that specific environment, you don't get it with the genome alone.
But if I can say something, yeah, or perhaps go ahead and then. Yeah.
I wrote a question about this. Can we start to draw that pie chart just using genomics, or do we actually need the proteomics?
What, do I have to draw a pie chart? No, I mean...
You are the pie chart guy.
No, the proteomic fractions. Yeah, but this is exactly the point, and I think it's related to the question about sensing, right? So that the pie chart is a proteome section, so it's actually what you're doing, yeah. But then what is challenging and is related to sensing is how do you take this decision and arrive to that balance?
But maybe I didn't. Sensing, I thought it was like
I think that
navigating gradients.
No, I think sensing is how you take decisions as a cell. Both. So.
Because I use the sense. I use the sensing of nutrients just an example about how decisions in the cell, and then like strategies in how to relate gene and strategies in these two different environments.
But related to this, I think that there is a point. So if I understand correctly, your intuition is that if you are in a good environment, so an environment that is rich of resources, you will need less to regulate or to take decisions, while an environment where there are low resources, it's more complex. But I think that typically environment, if you have, I mean, if you are a copiotroph, the type of environment you are exposed to is an environment where you have a lot of cycles. So your strategy is to be able to grow fast when there are resources and to deplete them as fast as you can. So the idea, at least, is that the typical environment that a copiotroph experiences is an environment where resources arrive in high abundance, you have to grow fast as you can, deplete them. And so to be in this environment, you have to take a lot of decisions. You have to find these resources, so you typically are motile, while oligotrophs thrive in environments where you have like low concentration that are low all the time. So you want to be efficient and you are not very flexible in things. So I think this is the dichotomy, right? And in that respect, the sensing come in.
I thought you were talking about like chemotaxis.
It's related. Right?
Yeah, but
yeah, but it's related in the sense that
regulating is different from sensing.
Okay, decisions. Yes, so it's the
navigating a gradient. You need to be motile. That's why I wrote the line.
Yeah.
So that was.
But there was something else I wanted to say, but I forgot. So.
My question or commentary is more towards what Nicole was talking about, the division between oligotrophs and copiotrophs. And there was this thing that it also depends on the environment, if it depends on the bacteria itself. And in one article is the big one, I forgot the name, it talks about mechanistic, yes, it talks about how the transition between being an oligotroph and being a copiotrof needs to not be very easy, so there is a selection between, it's not easy, and in this case you have not competitive exclusion, because if you had this passage between being a oligotroph and you don't have co-evolution, co-living in the same place, and also between what Martina was talking about, the variance between being a copiotrof and an oligotroph. Since in the article, the one about the trophic, then the nutrients, they used only 2 bacteria, and I think that is, we talked about that, about the extrapolation and how, I don't know, I didn't read the one, but just by talking with my colleagues, it felt so small, even though they chose the ones from the edges. They only chose pre-living, for example, and there you have the parasites and the others, they're symbionts, and okay, those types of the different functions that they have by being a copiotroph or a oligotroph in these, by being a parasite, are not represented in the okay, it's back. And that is just some thoughts that we have, that it's not a very good approximation, and maybe some of the things that we've been talking before that doesn't make sense, that are counterintuitive, it only works those 2 bacteria and may not be extrapolated. It's just a general thought.
Just to comment, they use these two model genomics, but they say they use also 32 additional genome sequences. So it's not only 2 at least, but yes, I agree that 32 is still seems like a small number to conclude all these general profiles.
So, but the question, if I understand correctly, is how much the idea of a dichotomy is. So there are 2 questions, I guess. One is whether it's really a dichotomy, and how much diversity you have within each group, right?
Maybe I didn't make an actual question. I just commented.
No, it's okay. It's okay. It's just to sort of frame it.
I guess maybe one question is: can we actually conclude something by only comparing two model organisms? Like, can we say like all copiotrophics will behave with this kind of profile? And I guess it's something that line.
There is a more recent article on PNAS, and instead of looking at like all possible genome features, they looked at the codon usage bias, which basically usually is thought as a good proxy for growth rate. And based on that, they had many more genomes, because it's like 10 years later, and so more, 12 years later, so the number of sequenced bacteria increased. And they make a point that they can separate oligotrophs and copiotrophs based on just on the doubling time estimated from codon usage bias. Which to me is, again, so there is this, my personal opinion is that there is this dichotomy, especially in ocean sciences, and it's very engraved in the literature. And so everything is trying to fit this big picture that started with 2 species and 32 genomes. But again, probably the thing to me that what I get from all these studies is that there is a continuum of species, and that's the thing that we need to remind. But yes, so the literature is still working on this, because there is this very strong idea that this is the good way to look at things, and that you need trade-offs to have coexistence. But this is a view that is shared. It's not the only one, of course, but yeah.
No, I think that is the point. But that somehow, I mean, the question is that is true. So if there is a dichotomy of niches, right? So that in some sense you have 2 niches that are filled by these two group of species.
I think it's a good idea. You are in the more traditional, in quote marks, ecology. You have this idea of this trade-off between the R and K, and it works relatively well, depending on the scale that you are looking. So I think the idea it's good, and maybe the only question is like gather enough data to build a good set of markers that you can use, and be aware that it's a generalization, as there are many others in ecology that work well in certain scales.
Yes, so yeah, I guess that a question is how you can also put these, what kind of things you can extrapolate from these, right? So what you can actually put into a test and derive from this dichotomy. I mean, in my view, there are sort of 2 things, right? There is a question which is more the why, so why you arrive to such a division of strategies. So it's like basically related to this streamlining theory. And one is what are the ecological consequences of this division. So
when I first, I started by the article which is not of the quantitative model, the one which is describing with genomic data the trophic levels, and when I was reading it, I was having a lot of trouble with the definition in a similar way that Nicole and Isabel said, because I was wondering, because the distinction between autotroph and heterotroph is a very clear one, and yeah, that's true. That's not, but it is more. That's not true. Yeah, but that's clearer than the distinction that I managed to find between copiotroph and oligotroph, because at first I thought, that they are in different environments with different carbon availability, but then they are not necessarily in different environments in like the scale in the scales we measure, but perhaps sometimes they are just taking advantage, the copiotrophic ones, of the heterogeneity of the environment, and the other ones are not really looking at the heterogeneity of the environment and are more concerned with the homogeneity and how to take full advantage of it. So I can see the dichotomy coming more from the fact that you can try and always keep taking chances and the like. So some game theory approach would perhaps clarify this for me, why that this chasm appears between such two strategies. Because if it was just a matter of concentration of availability of nutrients, I'd expect more of a continuum of species, or even a single species which developed a mechanism to take advantage of the varying levels of concentration of resources and the like. So, but in this case, from what I could gather, well, like when there is a new source of carbon which came, I don't know, from seismic activity or the like, you have an event. So there is a temporal asymmetry and there is a spatial asymmetry. And some bacteria are dealing with most of the space as it is, and others are dealing with this asymmetry that bursts and then trying to survive when they have to deal with the environment they are not really most fit to. So, but even then, if you are considering, you have many oligotrophs and not necessarily just one. So there are other in the sea. So there should be other factors besides the factor which was considered in the quantitative model, which is the ABC system, which I do not understand very well how it works, but. So I didn't feel very convinced by the way that paper tried to say oligotrophs and copiotrophs arise out of this.
This makes me really happy.
No, but yeah, I think that there are several points, right? So that you raised. Yeah, you want to say something, or there was another question?
If there are other comments on this.
I mean, if I can say something perhaps naive, I think that the dichotomy arises because of human nature, and because the bacteria that we were able to study in the lab are not the bacteria that are super abundant in the ocean, right? And so you say the bacteria that I can grow, I can study as much as I want in the lab, but then I go in the ocean and I see these things that is super abundant and that it doesn't grow at high concentration of carbon, and it's a mess to grow, and so then I have this dichotomy between what is in the lab and what is outside. This is one thing about the origin of the dichotomy. Then, I mean, I don't think that these sort of the dichotomy between high nutrient concentration and low nutrient concentration, to simplify, or stable environment and heterogeneous environment, are the two only limiting factors or the two only niches, right? So you can imagine that you have different metabolic preferences within each of the two classes. I mean, you can imagine you have different metabolic preferences between the two classes. You can imagine that other limiting factors are important within these two classes, so like predation or whatever else could matter, right?
Yeah, there is this thing that you mentioned. There is mostly one axis about nutrients, that is concentration, and then we forget about the diversity of the nutrients that you can have. And this is an axis that in this, for example, dichotomy is not included. And to me, one thing that it's really difficult to reconcile in my mind is that there is another way of classifying bacteria that is between generalists and specialists in terms of resources. And you basically have the same predictions in terms of genome size. You have generalists that can take advantage of many things and in principle have larger genome sizes because of this, and specialists that instead have small genome size and they can only do one thing. for example, with SAR11, for me it's very difficult to reconcile this vision because if you think about the transporters, the ABC, they're not specialists at all. So they can take up many things, and what sets there is the concentration apparently, but again, also that is not super clear why at a certain point, why when you have high nutrient concentration, they don't, I don't know, I don't even know, they don't grow. It's unclear whether they die or not.
Obligate oligotrophs that don't grow at high concentration of carbon.
So for example, there are these two axes of the way to classify bacteria that to me clash a lot with this type of definition based on transporters and based on metabolic pathways. Just to add about this.
No, other point that we were discussing was about when we talk about the streamline the thing, some organisms tends to lose some genes to get a smaller genome, and that we have two types of organisms, the free living ones and the ones who are symbionts. How can we try to figure out how is the network of these, for example, in the symbionts, we have these organisms that are the helpers, and the ones that can actually lose these genes because they can get these functions and these nutrients from the others. So how can we start to construct, to map these networks? And we are discussing like maybe there is a center organism that provides for everybody, so we have this main helper, or everybody is kind of helping each other. How is the structure of this network?
I think you can see it in, it can be both, but so I think there are studies trying to, I'm trying to understand this, for example.
Do we have any idea, or is something that we can,
no,
we just don't know
so
yet?
For example, let's say, if you have a very large polymer, like cellulose, the distribution of enzymes that can degrade cellulose, basically chop cellulose in small pieces, is not uniformly distributed. So you have few species that can chop cellulose, can chop alginate if you are in the ocean. And so basically if you want to take advantage of these big polymers, there are these species, and that, for example, you can call them degraders. So if you don't have the degrader, probably, and cellulose the only thing that is available, then the whole community that relies on the degrader. And this is the concept in ecology of keystone species. But actually, when you try to look into the, let's say, the distribution of keystone species in communities, it's very difficult to understand. So sometimes you don't find many of them. And the reason is that you don't necessarily need something like cellulose to initiate a metabolic chain. So you can actually use other simple, so in principle you don't need a polymer to have a metabolic chain. You can have a small molecule like glucose, and the way then trophic interactions start is because cell leak metabolic byproducts, and so there are other cells that can eat them. But the thing is that it's less hierarchical, so it can be a network. So I would say it's not, it can be both. But it's not so easy when you are in other organisms where trophic chains are really easier to understand. With bacteria, it's more complex.
One thing I was hoping to hear the other groups about, because I thought they perhaps had gotten to this point, is how the Black Queen strategy, which people from the other papers explained to me, would play into this. Because I wonder, it means that some organisms can shut down some functions depending on others supplying it, but I do not manage really well to connect how that may be a speciation, have a speciation effect, and produce those those divisions of niches between such species, because I can see some of them taking advantage advantage of others supplying their needs, or and diversity appearing within a community, but then suddenly, well, I don't know how I have the feeling that the populations may collapse when this
Tragedy of the commons.
yes.
Yeah. Well, actually, I mean, when you so you want, yeah. So the point is that when you have like you have to add something to
No,
this. Yeah.
I'm not gonna answer him. I just wanted to add that I had a similar question, and for me, what it sounds is that you're selecting something that is not this part of the population, the helpers. They're being selected even though they're not. Those traits are not good for themselves, and that's how. Another thing that I wanted to ask: how to maintain that? How does it does not collapses?
It seems to me that there should appear simultaneously two features, because their interplay may generate a stable separation. You need to somehow generate an advantage for at least some of the helpers, of the ones who take the burden, and you have to and you need the advantage for the other ones. And if you have this I think of two graphs of benefit like this, so and those curves are somehow mirror images of each other, somehow I can see successful speciation and maintenance of the communities coming out of them. But I didn't see in the papers that try to explain copiotrophic and oligotrophic development, and how some of them have less gene, a much smaller genome than the other, how that could have, at least a story of how that could have come up.
I think it's another question, but I mean, related to the tragedy of the commons, I mean, typically the scenario we have in mind is a scenario where you have a population at fixed size and you look at the fraction. So, but imagine like this example here. So what happens is that I'm not going to draw anything because it's going to be much worse than this. I can do this, but so typically the breaking down of complex polymers, for instance, is done outside the cell, right? So you secrete the enzymes, then you break down, and then everyone can eat them, right? Can eat the byproducts. Now, in this scenario, you could naively think of if everyone loses the function, then the population collapses, and that's it, right? How do I avoid that? Yeah, the cheater strategy. The idea is that actually you have a feedback because, I mean, of course you pay a cost, you have an advantage of losing the function, but it's also true that if the function is very rare, okay, only the ones that do that function are going to benefit from it. Because, for instance, the enzymes are going to diffuse at a constant diffusion rate, so they're going to be more concentrated close to the cells that produce actually those enzymes. Okay? Yes. Or you have privatization of these public goods, right? So the point is that the public goods are not. I mean, the idea. I mean, and this is if you want, is a theoretical explanation, but the idea is that you have when the public goods are not equally distributed. So if the public goods are very abundant, then you don't care because you're not limited by the amount of public good. When the public good becomes rare, you are limited by the presence of public good, and the producer of the public good have an advantage. So that's what prevents from collapse. But the idea of the streamlining is that you have actually a selection, so that if you look at what you pay in losing that function, so the fact that these public goods are not readily available to you because you have to be close to a cell, so something that is related to a cell that produces it, so basically what is the chances that you have that the byproducts of the degradation of these complex polymers, let's say, close to you, this is like what you pay, but then you have an advantage because you lose that function, and keeping function is, of course, constant. Okay? And so this benefit and advantage is not something that, and this is what is at the origin of the tragedy of the common, fixed, independent of the composition of the community, right? It's something that has some density dependence that then creates stable composition based on let's say, cheaters and degraders and cooperators. Thanks.
There is another thing that is related to what you were saying before, is that so if you forget about homeotroph oligotrophs, you only look at genome size. There is an idea that if you are in a that fluctuating environments, as you said before, and stable environments favor the two different strategies. So having a lot of genes allows you to withstand changes in the environment, while if you don't have them, you have to stay in a very stable environment. And for example, there are some papers that propose that the evolution of generalists especially is actually driven by these changes in the environment, meaning you have periods in which you fluctuate that favor something, periods that you're stable and favor something else, and you can have a spatial distribution of temporal distribution of this type of selective forces. But there is a paper, now I don't remember the author, that is about the maintenance and the coexistence of generalists and specialists that are defined by the genome size.
The other question I thought of was that in the case when we are studying ecology ecology slash evolution, I don't know, of microbes, you have a feature which is not present in when you study animals and the like, which is horizontal transference of. And I didn't read anything about it, but I was wondering if in the development of such things, horizontal transference would have a heavy weight, because if you amongst the population have individuals which have the dove strategy and individuals which have the hawk strategy, when the scenario changes, perhaps the diffusion, if the important genes for such functions are motile, you will have somehow a sort of transference in this process. And I think also that drift has to play a huge role, because if you had simply a homogeneous scenario with a lot of bacteria and occasional points with more nutrients, it does not seem like the scenario in which this kind of trade-off would really take place, because it seems like you have to sometimes get lost, and then you need to be able to metabolize the resources by yourself, because there won't be no. Then again, it's the trade-off. It's the thing of homogeneity. I forgot. Never mind.
About horizontal gene transfer, not all the functions can be moved. So if you can move, like, for example, one thing that is mobile are carbon preference. So it depends on how deep is the trait in the phylogenetic tree. So for example, photosynthesis doesn't move. It's very deep in the phylogenetic tree. But for example, carbon preferences, so the ability to degrade something, is pretty shallow. So this is something, for example, that can be moved. Another thing that is super common are antibiotic resistance. So this is one point about horizontal gene transfer. But actually, to kind of to increase the doubt, they find that there is a lot of horizontal gene transfer, even so you can find imprints of horizontal gene transfer even in SAR11, which should be counterintuitive because he's losing stuff, so you shouldn't exchange. So just to add that the picture is very complex, and this is a way of looking at it. But of course, you can find many ways to say, okay, this is not really, this is counterintuitive with respect to this other aspect that they find. Or phages, I mean, there are, so.
No, to stop.
Yeah.
So it's 1, so.
And you guys have to go?
We have to, exactly, so we have to stop now. So we'll see you next week.
Yeah.